All right, guys. Welcome to our third lecture of the semester. Thank you all for being here, especially because it's fall break right now and a lot of people are off going home or doing other unscrupulous things. So appreciate it. Tonight's speaker is going to be Dr. Ligia Coelho. She's an astrobiologist at the Cornell's very own astronomy department. She's very cool. And her fun fact for me to tell you all is that she sent the first menstrual cup into space, which is pretty cool, I'd say, and pretty unique. So I'll stop rambling to the mic now and I'll pass it over to her. Thank you guys again.
Thank you so much for the kind introduction. Also, thank you for being here. Thank you for the invitation. It's always a pleasure for us to talk about our results and our work. But most of all, it's very enriching to me every time that I'm able to explain them and to get the feedback. And actually, usually on these conversations, I have new ideas because I can see results from other perspectives. So this is also very useful to me. Feel free to ask questions. And yeah, so let's talk about astrobiology. So before I start on the science, and there are going to be just 2 moments of hard science in this talk that we will have to deconstruct what I'm showing. So just bear with me on those and I'll let you know when we arrive to them. Actually, I think you immediately see it because of the poor graphics I'm going to show you. But before we get to that, I would like just to talk about a bit of my background because sometimes it's confusing to people. I indeed work in an astronomy department, but I'm actually a microbiologist by training. My background was all microbiology, and I was actually under the impression that I had a very comprehensive microbiology training because that's what the university sold at the time. Oh, you're going to touch all the sides of microbiology. But actually, with time, I realized I was trained as a clinical microbiologist and not at all as an environmental microbiologist. Which, when I decided to start learning astrobiology and studying astrobiology, was very clear I was not I didn't have enough training for it. So it was also very normal that after that clinical microbiology training I ended up doing cancer research for a while. And also I developed medical devices for women. I had a startup for a while, and then I got this nice PhD grant that allowed me to pick what I really wanted to study. And I was not happy doing the biomedicine. And I thought, well, what I really want to do is search for life in other planets. And I had the opportunity to do that, which I'm very grateful for. So I went from a cancer researcher to a polar microbiologist / astrobiologist. And what I was doing in the poles was looking for data that I could use to help missions that were trying to find life on icy worlds that orbit Jupiter and Saturn. And we are going to talk a bit about those. And with that, I actually ended up in an astronomy department for my postdoc right now here at Cornell, working in an interdisciplinary team that is very welcoming to people like me that have a different background and different training. And I'm able to do my research interests, which is biosignature in exoplanets and biosignatures in icy worlds. And this is what today I'm going to show you. But as a side job, I really like sending stuff to space, sustainably, of course. And I really like to advocate for women's rights. And together, putting these 2 things together, we sent a menstrual cup, the first one, to space. And the objective with that was getting some data on the robustness of these medical devices in order to see if they could be used in space in long-term missions. But the most important thing with this launch was to launch the conversation, because until now, and continually, women in space don't have as many options as we would like them to. And for a long time, this was not due to the lack of alternatives, but instead to the lack of knowledge of the people that decided on their health in space, the lack of knowledge they had on these medical devices and alternatives for women to menstruate in a different way. So yeah, this is what I do as a hobby. And today I'm not going to talk about it, but feel free to ask me questions about it. I would like to kick off this talk with this sentence that is one of the main motivations why I do what I do. All civilizations become either spacefaring or extinct. This is a Carl Sagan's quote, and I know that when people see this sentence, they usually think about space colonization, which is an obvious assumption immediately when you read this. And most likely, he was also talking about that. But when I read this sentence, it tells me it's something different. It tells me something, it told me something that I found interesting and that I felt I was needing so I could truly understand life and biology, which is I need a duplicate so I can understand what's happening in this single sample we have. If we have a second example elsewhere that we can study, a second example of life, we will find a lot of the missing questions, the fundamental questions we have still about life on Earth. And that's my motivation. And that's what I think about when I see this sentence. Actually, there's a thingy. Oh, there you are. So astrobiology is a multidisciplinary science seeking to answer some of the questions, these fundamental questions. And some of these questions are, how does life begin? So origin of life. What is the future of life on Earth and beyond? And if there's life elsewhere in the universe. So the first two are not at all something I'm equipped to talk about. So some people think that astrobiology, we all work on these three things simultaneously, but that couldn't be further from the truth. People that do origin of life usually do things much different than people that look for biosignatures or technosignatures. Technosignatures are signatures of evolved life, let's say. People that they are studying that could be studying what is the future of life on Earth, for instance. People that study biosignatures are people that study this third option here, which is to answer if there's life elsewhere in the universe. And that's what I do. But of course, if you have any question regarding the first two, we can have a discussion among people that, because I'm not an expert on that, and we can just have a nice discussion on it. I can share some ideas, but I'm not at all equipped to talk about them as an expert. And if you want to really think about life elsewhere in the universe, still there are two types of research that you could do. You could search for life in the solar system or outside the solar system. And this also couldn't be more different from each other. If you're searching for life in the solar system, you are obviously not searching for a civilization, because we are pretty sure we kind of are the only ones in the solar system, if you can call us a civilization at all. So what you are looking for are microbes, are a microbe world, single cell organisms, maybe even. So there's a limitation on what you can find, a very, very big limitation. You are narrowing your research for just one type of life forms. But there is multiple machinery you can use to actually do that, because you're close enough that you can send missions to probe or to even land on planets and moons in our solar system to search for it. You can use much more than a telescope. You can use them too, but you can actually use a spacecraft that approaches and maybe lands and drills and make DNA extraction or cultures even. Now, if you're looking for life beyond the solar system, you don't have the first limitation. You can be looking for any type of life form because there are no limitations, no rules. You could have civilizations, even more civilized than us, hopefully. Or you could have plant worlds, or you could have microbe worlds, but from different flavors even. So there's no limitation on the sampling, but there's a big limitation on the machinery you can use. Because you're so far away, you can only use telescopes. And there's something that's never going to change. You're never going to get closer. So the approach and the reasoning has to be much different. And the type of biosignatures you look for, unfortunately, also have to be much different. Sometimes these two fields touches, and I'm going to show today an example. So let's start with the solar system. My actually my work as an astrobiologist started in the solar system, and then it took me elsewhere. But in the solar system, my main objective was to find some place here on the solar system where there were the main requirements of life, and that I could, if they had the main requirements of life, I could create data sets that could be used to find life through missions or guide the missions that were going to these places trying to find life. So I would like to ask everybody here if they could share with me their idea of what is the main requirement for life before we continue. Energy. I can tell you're a physicist. Water. Who said it? Correct. Sorry? Did you say coffee? Because for me, it is a main requirement. Carbon, exactly. So you all touched the three components already: energy, water, and essential chemistry. Liquid water elements where carbon is included, and energy. And these are the main requirements for carbon-based life. We can talk about other types of weird life that are theoretical, but very important and very impressive studies on them. And those, of course, will extend the places where we can search for life. But if we are only talking about life as we know it, there are some places in the solar system besides Earth that we could theoretically search for. For instance, the icy worlds, the icy moons of Jupiter and Saturn, these small moons that are covered with ice, a thick layer of ice that is on top of a big ocean of liquid water. And this liquid ocean, and this is one of the most important parts, is in contact with a rocky core, which means there's going to be a lot of chemistry going into this ocean. And the fact that you have the ocean and that liquid form also tells you there's a lot of energy there. So you actually have the three components right there. You have the chemistry, you have the energy, you have the water. You have even more water than on Earth. And these moons, so far, the ones that have these three requirements are Europa, Enceladus, and Titan. Enceladus and Europa are by far the ones that have much more attention. And if we have to pick one, Europa is the one that so far has had more attention by the researchers. So this actually tells you that on the solar system, the major habitat for life is not a planet like Earth. It's not Earth. It's icy environments. If we go by coverage, what we consider habitable in the solar system, the majority of that space, the majority of that area, it's ice. It's an icy biome. And that's what's winning in terms of numbers in our solar system. And we haven't even found life there yet. Or maybe we will never, but it is habitable if we follow these rules. If it's habitable, we have to consider it's by far the major solar system habitat. So I would like to take you all fast forward a week from now here in Ithaca. And these types of landscape is what you're going to find. You're going to find a completely covered surrounding. Enjoy the good weather while it lasts, because this is coming for you. So instead of thinking of Ithaca, you can also think about the Arctic, because it's very similar sometimes. And my research, following all these reasonings, brought me to the Hudson Bay. So I did field work on Hudson Bay. And why? Because Hudson Bay, it's a very particular and undiscovered place by researchers. Although it's very big in the Canadian North, and although it has the fastest rate of sea ice loss in the Canadian North, it wasn't studied biologically since the 50s, before I touched it. And the reasoning for doing it for astrobiology purposes is that besides being a cold environment, it also was ice covered. Just a few, it wasn't covered, the bay is not covered by ice for some parts of the year. So there's a transience on the ice coverage, and you'll see why that's important. It has also freshwater inputs around. So it means that there's going to be gradients of salinity around these waters. And the most important fact is that although it's very cold, as cold as the high Arctic during winter, because of a polar front, a whole thing, we can talk about it, but I'm not going to go as much into it. The fact is that this is still a subarctic location, which means it has more radiation than the Arctic. So you are going to have more radiation, but as cold as the Arctic. And that's a very interesting combination for astrobiology. So here I am going to Hudson Bay, a Portuguese that have never seen snow before. Until this day, I'm not joking. The first time I've seen snow was in the Arctic. Go big or go home, right? In the middle of winter. And I got a taste for it because then I moved to Ithaca. And so it's my first day and I'm doing my first sampling. I'm a first year PhD student. And I get this metadata where I see that the place I'm sampling is ice covered, looks good. Has an ice cover of 1 meter thick. Why not? Water salinity and water temperature are this and this. Okay. And then I'm like, okay, can I compare these to other places on the solar system that are optimal? Let's see. So if we compare to Enceladus and Europa, that are very different, that was my first mistake, putting them in the same box. We can see that they are both ice covered. I'm happy already. But then the ice cover is dozens of kilometers thick. And I'm thinking, I might as well just give up because my place that I'm sampling just has 1 meter of thickness of ice, which means the pressure is completely different. The biology has to be completely different. Not so fast. So the fact that you have dozens of kilometer thickness of ice doesn't mean that you don't have water, liquid water, that is through this ice, that is perched in this ice. In fact, that was proved that exists. You have perched water bodies that are less saline than the ocean underneath in the ice shell of Europa and Enceladus. And until this point, nobody had found a planetary field analog that would actually mimic that pressure and that salinity. And Hudson Bay is actually one of the best places you can use to do that. Use is a bad word. You shouldn't use biomes on Earth, but that you can study for that purpose. Because you have a fast sea ice loss and creation. So it's transient ice. And you have less saline water than in the Arctic Ocean because it's a bay. So it's actually, things were shaping up, but it was a very harsh first day. I'm not gonna lie. And then water salinity and water temperature are the same because it's the same laws of physics. We are in the solar system. It's going to follow the same laws of physics, thankfully. So I was convinced I was studying a planetary field analog to icy moons, at least for the perched water bodies that were on that ice shell. And so I went on with my sampling. So here you're seeing 4 sampling locations. And those sampling locations are following a salinity gradient because, oh, I'm sorry about this, a salinity gradient, because we are close to a river here. So on site 4, we have decreased salinity. We have almost fresh water, almost 0%. But on site 1, we have the highest salinity that we sampled, which was 1.5%. As a reference, the ocean is usually 3.5. So we weren't still on a very saline location. But again, I'm not studying the ocean of Europa. We are studying the perched water bodies, which are more freshwatery. So every time you see 1 means a lot of salt, 1.5%. Every time you see 4 for the site number, it means less salt. And in between, it's following a gradient of more to less. And so my first question was, how many microbes can I find on this ice layer? Because it's looking pretty boring to me by now. It looks like I'm alone in this bay, except the guide. Of course, we had a guide with a rifle and all because polar bears. But it wasn't looking like I had a lot of life around me for me to study. It was getting me a lot of anxiety, to be honest. So I was like, okay, let's see how many microbes I can find. So some of you know this joke. Please do not say anything. I would like to know how many microbes you think we can find in this landscape. Just throw it out there. Best guess. I'm a patient person. Am I hearing 1,000? Who gives more? Come on, people. I would love to write that on the paper. Well, it doesn't have to be precise, but order of magnitude. Thousands, millions, more than millions, less than 10 millions. Okay. We have 3 and we have millions. Anything in between? Anybody want to throw out? Thousands. Okay. Well, we have 3, we have thousands, we have millions. I think I can continue. The person that said thousands is correct. So it's not millions or 3. Per milliliter of ice. So it's actually millions. So we actually have thousands, but per milliliter. So if you melt this ice per 1 milliliter, you'll get 10,000 microbes. So it's even more than millions. It's an incredible amount of microbes. And incredibly enough, you look at that landscape, what you think is it's actually sterile looking. It's boring. But per milliliter, you have 10,000 microbes. This is what we found. We found, of course, that the water underneath these ice had much more microbes. Apparently, even though a lot of them like the cold, the ones that doesn't are still more. And we found that we would increase the number of microbes as we were approaching the river. So it also looks like salinity, as much as the cold, were selecting against some of the microbes. You can see this here. You can see the number increases in the water microbes as we are reaching site 4, which is less salinity. But in ice, we don't see that happening. And this is the ice that is on top of this water. This water and this ice are the same thing. The only difference is that it gets cold, it freezes. So it's the same original material, the same original sample. Why are we having this difference? So we see that in ice, as we approach the river or not, doesn't make that much difference as it's making for the water underneath. And that was curious. And so understanding more these locations, we see that actually on site 3, that it's pointing there, we see actually an anomaly happening where there's a stupid increase of microbes out of the blue. That again is not making my story very easy to tell because it's not following a gradient. So what's happening? Looking at which microbes were there, and we are not going into that. And this is the first bit of hard science. You can see by the fact that you cannot understand this graph. And it's not only you, it's my colleagues as well. And me sometimes. I always have to go back and read what I wrote because I look at this and I'm like, but this is metadata means, this is big data. It means it's very hard to plot. And we want to really convey all the information, but it's not something that you look and you immediately understand what's going on. So what's going on here is that, I don't know how to use this. There you go. What's happening here is that, just look at the first one. You have salinity decreasing from that side to the other side. It means that on the first 2 bars, you have ice from site 1. And then on the next 3 bars, you have water from site 1 in triplicate. Then you have water from ice from site 2, water from site 2, also in triplicate, water and ice from site 3, the same way, the same trend. And then on site 4, and this is why it gets also very confusing, I don't have triplicates or duplicates because it was darn cold on that day and it was hard to sample. And it was very okay for the referees that reviewed this paper. So I hope it's okay enough for you guys. But that is why it's very asymmetric. But anyway, so what you can see is that every 2 bars that are similar, next to it you have 3 bars that are similar. Then you have 2 bars that are similar. Next to it you have 3 bars that are similar. This means that ice and water, ice duplicates are similar between each other, ice triplicates are similar between each other, but ice and water from the same place have nothing in common. You can see that for site 1, site 2, site 3, more or less. But then site 4, ice and water, look more or less the same. So what we are seeing is that for sure salinity is making a difference, because the only differences between site 1 and 4 so far is salinity. But then we look again, and here it is, site 3, the weirdest of them all again. So something is really messing up the communities on site 3, the communities of microbes. It showed on the number of microbes, it's showing on the type of microbes. Something is really making a difference here. And so when this happens, one has always to second guess itself. Like, did I contaminate this? Was this a bad day to sample and I didn't notice? What happened? So one should always go back to the past, and the past means your notes, and your notes means the metadata, the data that you took that day about the weather, about the probing that you did on that lake or on that ocean or whatever, or the snow depth, all these things that you should always put on your notebook, because if you're in the middle of your paper writing and you see that something doesn't make sense, that's just going to save you. And I went back to my metadata. And I saw that on site 3, we have more chlorophylls, which is a pigment that make photosynthesis. You have it on plants. You have it, oh, hello. You have it on algae. You have it on cyanobacteria, not only plants. And you have a lot of biopigments of cyanobacteria. So for sure, what was happening is that on site 3, we are having a lot of photosynthesis. That also coupled to the fact that snow depth, as you can see on site 3, is less than the others. It means that this ice was more exposed to radiation, was more exposed to light. So we were having more production, more biomass production through photosynthesis. And I was thinking, well, can I see this through my cultures? And yes, I could. So from this very boring looking third location ice core that you see here, like, how could I know they were different from each location? Look, you can, like, this is crystalline ice. Like, how could you tell that this ice would actually have these colors growing inside, but microscopically, meaning we couldn't see or we couldn't probe unless we grow them to a point they are so enriched that we can see. So this is what's happening on site 3 microscopically in terms of life. They are filled with colors that we can see, but maybe we can make our machine see. So biological pigments, now that we are understanding that this is where I'm going with, biological pigments are substances produced by living organisms that will have a color resulting from selective color absorption, selective light absorption. And they will be very, very characteristic through spectroscopy of all kinds: absorption, reflectance, transmission, fluorescence, whatever you decide to use, they will always be weird. And they're always going to be something that diagnoses them on the spectrum. And why does life produce biopigments? To mate, to fight enemies, to thrive and produce a lot of biomass, or to show the colors of their heart. Go Cornell. Basically to survive, right? So it protects us too. We pigment. You can be even more pigmented if you eat oranges and you put yourself, carrots, sorry, and you expose yourself to the sun. Not that I'm telling you to do it, but you totally could, and it's reversible. We all pigment, and almost all life on Earth pigment if it's in the surface. And this is because it protects us against radiation, dryness, lack of resources, changes in temperature, basically weather forecast in space. Which means they are important biosignatures for us to look in space too, because they are clearly linked to ice biomes. If you're in ice, you must have pigments. We actually reached this conclusion by comparing the water underneath and the ice, because they come from the same biome, the same sample, the same origin. Why would the ice be so different from the water? We actually reached the conclusion that it was because you see the bars in yellow and the bars in gray. The bars in yellow shows you the pigmented organisms on those ice cores, and the bars in gray shows you the non-pigmented ones. And you can see that you have ice, water, ice, water, ice, water, ice, water. You can see that in ice, you always have more pigmented bacteria than in water. The ratio between pigmented and unpigmented is always much higher in ice. And it's because ice is exposed to the radiation. So it means that on icy locations, even outside of Earth, if life is carbon-based, they better have the pigments. Otherwise, they can't survive. To our convenience, pigments are highly detectable remotely, because they are so bright, they reflect so bright and so distinguishable that we can very easily find ways to train our missions and our machinery to search for them. The problem is, however, that if a biome with 10,000 microbes per milliliter looks like this, we don't stand a chance to find the aliens. Because if I'm standing there and I can't see them, me putting a telescope and directing it to an icy exoplanet, it's not going to find it either. I'm much more far away. Even if the tool is so powerful, much more powerful than my eyes, the power of proximity, it's very hard to beat. And if I have the power of proximity and I can't see it, that's bad news. Except, luckily for us, an icy biome, it's not always like that. It's not always boring and sterile looking. An icy biome can be like this. You can find green beaches on Greenland. You can find pink watermelon snow on Sierra Nevada, and this is a microbe that makes this color. Or if you are having a nice cruise through the Pacific, if you look over the board, you will see that the water sometimes will bioluminesce through as the boat is moving and the water is moving, it will make this glow of bioluminescence. And this is actually observable from space. All of these examples are observable from space. So if we have a situation where we have exoplanets, where this is the rule instead of the other example, we can see life, or we can build telescopes to see life. We can maybe have an icy exoplanet that is purple, or orange, or green. It doesn't have to be the crystalline, boring looking, because that's only because on Earth right now, we have those biomes are conditioning to the high dominance, to the high competition for dominance that those microorganisms are actually doing with each other. Those examples of the colorful landscapes that you saw, it's because one of those organisms is winning that competition. On Earth, that only happens for some moments, but that's because Earth is like it is right now. It doesn't mean that before or in the future, Earth won't be more colorful or won't have different colors instead of green. It can be a purple Earth. Maybe in the beginning, Earth was actually purple. There are some papers about that. Also, it means that other exoplanets like Earth have no reason for them to be ice-looking like the Arctic or green-looking like our normal landscapes. It can be all other colors, and we have to search for them and prepare the missions for them. So the challenge then I had after I realized all these was that I had to convince my colleagues in astronomy to stop looking for the green. Because finally I said, yay, not only me, other people before me said, yay, please go look for biopigments. They are amazing. We can see them all around us and maybe they dominate exoplanets. Please prepare your telescopes for that. And they were like, yes, chlorophyll, we will find you. And I'm like, no, that's not what I mean. Sure, chlorophyll is great. Also prepare for that. But not only that, because even on Earth, we have situations where that's not the case, where the landscape is not dominated by green at all. We have photosynthesis that occurs and the biopigments are purple. Or the green, or can be green, but they can show different peaks on the spectra. Just because you see green and green and green doesn't mean it's the same type of green. We all see green differently even. If we use that as a science, we are doomed. We have to put some objectivity to it, right? We have to look it through spectra. Different shades of green will have different spectra. Different green from different bacteria will have different spectra, even if it's close to the same shade, because it's different biopigments. It's different chemistry interacting with light. We can even have photosynthesis that doesn't use light. It can use heat. And those will have different pigments, because why would chlorophyll be there if it doesn't have the light? So that was my challenge after that, was I have to write the papers to make my colleagues that are the ones looking through the telescope not only look for the chlorophyll, because that's like one example. And it's then the same problem. We are only looking for a type of Earth that maybe it's not unique, but not that frequent also. We have so many other options. Even if Earth is restarted right now, maybe the dominance wouldn't be green. This is Darwinism. Just happened. And there's nothing against it. So it doesn't stop happening. This is how evolution works. So this led me to my research to actually focus more on the exoplanets. And now that we saw how a research can be done for solar system, and remember that for the exoplanets, the problem was that the sampling was broad, but the machines were not. Let's see how we can actually look for life in an exoplanet. So instead of water, carbon, and energy, for exoplanets, we cannot use these basic requirements of life as our guide. Unfortunately, we don't have the power for that. The only thing we can use is if they are in the habitable zone or not. And what is the habitable zone is the Goldilocks, if you also know it by that name. It's the location on the system where the planet is positioned in the right place to maybe have liquid water, if liquid water is there, if water is there. So this is our concept of habitability. It's not anymore that has the energy and has the carbon and has the nitrogen and it has sulfur and it has energy from different parts. It's not that anymore. We don't have the power to resolve that much about exoplanets, unfortunately. It would be great, but we don't. So we start by calling them habitable if they are in the habitable zone, but mind you, we don't even know if they have the water. And then we can use methods like transit spectroscopy or transiting to identify the exoplanets, but also to identify, oh, it's gonna work, yay! To identify much more than their size and their position. We can identify what chemistry is on this atmosphere here, because the light of the sun that just interacted with the planet that you saw here made the molecules move in a way that the light that we are getting on our side will be changed by that. And we know how carbon change it, how hydrogen change it, how oxygen change it. We know that by our references here on Earth that we measured in a lab. So the light that we are receiving is already transformed, is already changed by elements, and we can trace that. So that first transit that you saw right in front of the star already tells us something. But then, and the one that I'm more interested in, and I'm sure I can do this. Yes. There you go. So the one that I'm more interested in, it's not as much as this transit here. Although some people do look for the chemistry here, I'm more interested in this second eclipse where you're seeing, oh, beautiful, you are seeing exactly the light on the surface. And I can directly see what's happening on the surface. So I could have the reflectance spectroscopy. I could have the reflection back to me, the light that is reflected back. This light also interacted with what was in the surface. And the moment it gets back to me, it's already altered by the molecules that are on that surface. And the molecules that are on that surface, if we have a nice reference library of all the possibilities, for instance, if the surface is covered with biopigments, if we have the laboratory measurements of those biopigments and the measurements of their reflectance, we maybe will have a nice catalog for us to go and search for direct imaging of these planets in the future with telescopes that are powerful enough for that. And so in order to do that, what I'm building now, it's these catalogs of life. I'm building on this work from Professor Kaltneger that already gave a talk here also, I think last year, that in 2015 started his work and created the first catalog of life where they grew and got the reflectance spectra of several different organisms with different pigments. The idea here was not to do this for a specific environment or a specific organism at all, was just to prove the concept of the methodology that it worked. And then Ligia went to the Arctic and saw ice from the first time, not ice, but snow, and got all these colorful microbes and contacted US and Cornell and said, oh, hey, let's do the same, but for the icy locations. And here I am. This was part of my PhD, so I was also here in 2019 to do this. And I brought with me 80 microbes, not directly with me, because you cannot do that from Europe. You cannot bring biota with you. It's no, don't try it. Even using FedEx is complicated. Imagine if they open the bag and no, don't do that. So it was FedExed through the institutions like it should be. But yes, I arrived and I had my biota to measure in this amazing university, in this amazing setup. It was during fall. It was so beautiful. And then I saw snow again. I was so excited. So I used the laboratory here. I built this catalog of ice, life in ice. So the colors of life in ice to help future missions find these colors now on icy exoplanets. So again, I did the fieldwork, I did the microbiology, and then I came here to Cornell and I did all this spectroscopy where I grew again the biota, the bright colors, and we used reflectance spectroscopy in the visible, and then we created the database of all of these biota, and we put it to the public so that people that are looking through the telescope can use it. And there are people that are designing the telescopes in 20 years don't look back at me and said, oh, we have the telescope and it's in this wavelength that is completely useless to you. And I'm like, but now we cannot see anything still. And I have to wait 20 more years. Like my life is not, my lifespan does not allow for that. So yeah, so the idea is that this is done before the telescopes are finished, or even finish designing. And then what also happened was that by mistake, I left these filters for the first time that I did this experiment. We use filters so that the biota is measured in a disk or in a circle that mimics a planet. And we use a type of spectroscopy that's specialized for us to get the signature as if we are looking at a dot in the sky, just like a planet. So I did this the first time and I forgot the filters on top of the bench. And I only noticed that 1 week later. And when I noticed them and I was about to put them in the bin before anybody looked at and realized what I did, because leaving biologicals on the top of the surface is not a good thing. It's not good practice. And I was on a visa, so it's not something that you should do. When I was about to put them in the bin, like, shh, have this. Before doing that, I noticed how they were much more brighter than the ones I just measured a week before that were still full of water, fresh. They were drier. They were drying looking. It looked like they were not hydrated anymore, and the colors were much brighter. And so I thought, well, let's measure this again, but now drier, in the dry forms, to see if what I'm seeing, the machine can also see more reflectance. And we actually did. So what you're seeing here is all the biota divided by colors, and the spectra of all of them divided by colors. So in some graphs, you're going to have, in some plots, you're going to have 20, in another 10, in another 30. It's because some biota, the majority of biota maybe was orange, then you have a third of them that was pink, then you have just 10 of them that were green. They were divided like this. So you can see that. I don't know if I can move here, if that's okay for the people in the Zoom meeting. So as you can see here, this is the fresh samples, and this is the dry samples of the same colors. Here you can see the clear signature of the colors, which means that the biota have a diagnostic signature, which is already a good start. It means that they are really diagnostic, and if we maybe see them in the surface outside of Earth, we could identify them because they are unique enough. But then when you compare with the dry samples, you can actually see that the same signature, it's the same one because it should be, it's the same biota, but it's much more reflective. The reflectance, some of the peaks almost reach the maximum of reflectance that you can have. And this was very significant because it means that on planets that are not as hydrated as Earth, or it doesn't also need to be as dry as Mars, like an in-between planet between Mars and Earth that might exist in other systems, life is maybe easier to find. This is important because in the future, those telescopes will have a very big problem. There will be a limited amount of time to see, to observe. And because the planets we want to observe are Earth-like planets, they are very small. The necessity of time to be exposed to that location to see it, to receive enough light to see it, it's going to be abysmal. The time that we're going to need per planet is going to be enormous. So there's going to be a lot of competition, a lot of problems with scheduling. And if we can help them make a decision on the best planet to put their chips on, that's already useful information. Maybe people should start looking into intermediate planets between Mars and Earth. Maybe that is our best shot. Maybe not, but these types of work and modeling and these types of analysis that you can do on a database are quite powerful for that decision making. And this is what we showed. We also showed, and this was something I did for fun at the beginning, but then it ended up being an interesting result. I wanted to show to the astronomers how taxonomy wasn't important for the biopigments, because here we go with the chlorophyll. The chlorophyll is very important for photosynthetic organisms. It's usually the sign of bacteria or the microalgae or the plants. And that's very well for chlorophyll containing organisms, but organisms containing other type of pigments, actually, if it's this species or that genus or that taxa, it doesn't matter because you're going to have the same pigment on all of them. So it's not linked to a specific hereditary line or evolution line. Taxonomy does not have any power on these pigments. So we shouldn't be mining that. We shouldn't be looking for a specific organism. We should be looking for biosignatures that are linked to environments in general. So this was our way to show it. Because if I showed genes and proteins to the astronomers, they would be like, what? But if I show this in spectra, which is the language they can read, maybe I can make my point. So that was the objective. So the main points of all this work was that always keep track of contamination in your ice cores, because if you find something weird or interesting, you're never going to trust it if you didn't mind that. Pigmentation is important in icy biomes. I think this is the best and the most important thing I want you to today leave this talk with. Pigments are important on icy worlds even more. Ice has more pigmented microbes than water, and salinity and radiation will be the major drivers on icy alien biomes. Pigments are biosignatures of different environments rather than specific organisms. It doesn't follow taxonomy. Different microbial ecology in icy exoplanets will affect its detectability. And drier exoplanets are better candidates for observations because the pigments, if they are there, are going to be much more intense. And at the end, astronomers searching for colors, please, please, please. It's not always the green that wins. Don't always and only look for the green. On that note, the next paper that we fortunately and hopefully will have out is called Purple is the New Green, because we are showing a whole catalog of just purple bacteria, nothing else. And what environments are more, favor more this type of bacteria. So that planets that are prone to have those environments, for instance, planets around M stars, where the light is red mainly, maybe what we should look for is the purple or the pink and not the green. Not the green, ever. Just in these situations, but yeah, ever. And this work is on the framework of the work of Lisa Kaltneger. She's my PI. She started this idea that other Earths might have other colors. And then she was like, and this is something I really want to study, but I don't know how to grow stuff on the lab. So I have to hire people. And she ended up hiring me, and I was very happy with it. And I'm very lucky to be able to study this as my livelihood, study colors. All microbiologists secretly love the pigmented bacteria. And I have the ability to just live by working with them and ignoring the others. And I'm very happy. So she started this idea that we should look for alternatives to what we see every day. We shouldn't be biased by our own experiences and impressions of Earth. We should measure everything we can and model planets that could be dominated by it. And then she created the Carl Sagan Institute at the astronomy department here at Cornell precisely for that, because she understood that just astronomers couldn't do this work, or just biologists, or just geologists, or planetary scientists, or astrophysics, cosmologists. We all have to be together working on this. We all have to be hands-on, talking with each other and collaborating. But for that to happen, we have to have an institution that bridges us. Because if there's something the scientist doesn't like to do, it's talking with people we don't know. Or even the ones we do. So yeah, so we needed a setup, an ecosystem where that was a friendly thing we could do. We could meet for coffee, but also talk about ideas. And there's never a coffee, Carl Sagan Institute coffee that occurs that people don't end up with a new paper idea for them to collaborate with. Really, the only thing that it takes is for us to be together for one coffee opportunity. So yeah, so we are reaching the end. I'm seeing Ben also getting up, being like, get on with it. It's almost time. Yeah, so I would like to leave the talk. I started with the Carl Sagan quote. I'm going to finish with the Carl Sagan quote more or less, because this is not something, it's a quote of him, but he didn't invented it. It's a Laplace principle applied to space, let's say. So one of the things that motivates me is thinking that for us to be not extinct, we need to be spacefaring. But what motivates my work before I publish anything is thinking about this. Because I know how this topic mixes with emotions for a lot of people. So I want to make sure that what I'm putting on a paper is what I'm sure it's the truth. So extraordinary claims require extraordinary evidence, and this is paramount for astrobiology. Thank you very much. I'll be more than glad to answer to any questions, even about origin of life. Oh, cool. And for the person that almost said coffee. Not for a Portuguese.
Yeah. Okay. So you said that a third of your pigments were orange, and you have a whole paper about purple. What color do you think is would you think to find the most of?
That's a great question. I was actually hoping nobody would catch that. I'm joking. Yes, my first paper was more focused on a lot of different colors, not specific purple. And that is now the new line of research that I'm following. And then I'll jump to another color. Maybe I'll bring back the green. You never know. But that's a great question. And recently I was also asked not only what is the more important color to search for, but what would be, for instance, the color that is connected with the origin of life, for instance, which I don't like to touch on the subject. But there are colors that are more specific for certain environments and for certain planets and for certain times on the planet. There's a nice paper about how the origin of life is purple. Purple is the Archean Earth color, or Earth's color. I would say that if you look for an icy location, purple, maybe it's not your color. Dun, dun, dun. Most likely it's the orange and the pink and the yellow, because those are the colors of carotenoids, which is a type of pigment that is specific to make you just super strong against the cold and against the radiation. So logically, that would be what I would like to have if I was on that surface. And the colors, and it's interesting how the colors will change as the pigment will have a different purpose. And that's just so convenient for us. So yeah, it's a great question. I don't have the answer, but that's my guess. Of course. Thank you.
Yes. Oh, hello. If I'm remembering correctly, I think you said something to the effect of carbon-based life forms need pigments in order to protect from radiation. Does that mean that other non-carbon-based life forms could have something different besides pigments?
Who knows? Yeah, they could. Carbon-based life forms will need to have the pigments because on Earth, that is universal for all life forms. We have exactly the same pigment as yeast. Think about that today when you go to bed. The thing that makes your bread and your beer, if you're above 21, is the same, it has the same pigment as you, melanin. It's because it doesn't matter which species you are, we all have the same problem. We all have radiation, we all have temperature changes, we all have dryness to deal with, and the pigments are here to solve the same thing. So it means that on carbon-based life, you always will have to resort to the pigments. That's the conclusion we can have by analyzing Earth. If life is made of silicates, or if it's made of methane instead of water, my guess it will need a structure still that shields it for radiation, because I cannot see life that could be happy with changes in radiation. It will probably be okay with steady states radiation, but if you have flares or if you have an incredible exposure to radiation like you have on Europa's surface, for instance, there needs to be some shield. If that shield is just that life that is not carbon-based is always on the subsurface, that's a problem, but that's possible. If it's another molecule they make out of silicon, like a rock, rocks are made of silicates, maybe life that is made of silicates will have a shell made of rock. I don't see how that is different from life on the subsurface to us in terms of detecting it. If it's going to be colorful, maybe not. I don't know. Can I? Oh, yes.
So connecting with that question, do you expect like if other life is carbon based, that it would have the exact same pigments, and if it's different, then would you still be able to expect to like identify it using these same techniques and notice that it's a pigment? And if it's not carbon based, like with those others, then again, the same problem.
Yeah, that's a great question. That's the question of my nightmares, that every time I submit a paper, I'm bracing myself for that. If life is carbon-based, the best guess we have is we'll need pigments. If the pigments have the same shape as the ones we have on Earth, maybe yes, maybe not. We can constrain that in a line of research, which is called computational chemistry, where people could actually maybe make all a simulation of all the type of pigmented molecules you could have, and constrain in which type of chemical settings they would be more likely to exist. And maybe doing that, we will find new pigments we don't know about. I don't think that has been done. So until that's done, I will have reservations giving you an answer. My work is based on the assumption that maybe it's not going to be general like this, but at least we have to have other planets where this is the colors. And for this to be the colors, the spectra will have to be similar in a way. But then I think that in the future, computational chemistry will also need to pick up my work and see features, not features, but patterns I'm not. Because I'm being very basic about it. I'm like, does this does this, and this does this. Biosignature, biosignature, biosignature. And I'm not saying, okay, maybe the ratio between these two things and the steady function here, maybe there's a pattern that always appears in all the pigments. Nobody has done this. No, in biology, even in biologists, the ignorance about biopigments is astonishing. They just put it on a basket. I'm included, okay? I'm including myself in here. We put them in a basket of, they help us do photosynthesis and fight radiation. That's it. There's really no more developing on that. So yeah, we just don't know. I'm working with what we have at least and what is known. But the more I do, the more I understand how much we don't know. And I thought the databases were much better because I think everybody loves the pink bacteria in the lab. Why don't they just study it? It's crazy. I never realized they didn't. So yeah, maybe if you want to do computational chemistry, I would love to read those papers.
So I'm actually going to ask a question, and I'm going to sit at one of the tables so you don't have to look to the right. I'm going to sit here and look at you. So I guess you talked about like the pressure difference because of how there's kilometers of ice on Europa, for example, and also at the kind of seafloor on Earth, right? There'd be different levels of exposure to radiation from the sun and whatnot. So do you kind of foresee like a potential, probably a future because we're not quite there yet, but a potential avenue of research for you would be to look at microbes found at the deepest levels of the sea, especially, the ones that hang out near the I'm blanking right now. The
thermal fans?
Yeah, thermal fan. Thank you. The thermal fans. Do you think that'd be like a possible like avenue for you to explore? And you'd be interested in that?
The submission I wrote today.
Really?
See, you're in the state of art of things.
Wonderful.
It was not full on that, but it touched that already. I do have an interest in that because another misconception, and I touched a bit on the topic, is that all people think that photosynthesis needs light. But we do have photosynthesis happening on thermal vents, and it has pigments. And it's because it's feeding on heat. It's feeding on infrared, which is also radiation. Of course, the ones that we have in the surface will love the visible light because that's what they have. It's not a very good competition if you use infrared and you're in the surface competing with cyanobacteria that uses the visible. You just lost it. There's no way you can win. But if you go to other locations, cyanobacteria will be like, where's the visible light? It's not here. You die. And I live because I do the infrared. This is what the other bacteria will be doing. And there's a lot of, there's not a good reference database from those that can be used for icy worlds, even on the solar system, that have the deep vents and a big shield. And maybe potentially if there's life there, it will be similar because you always need photosynthetic life first. Or not first, but in some way for you to have a lot of biomass. Yeah.
More. Then we'll go to the Zoom questions real quick.
No, there's
So earlier in the talk, you were talking about different properties of icy moons, and I was wondering, like for Europa, for example, how do you know what the salinity of the water under the ice is if it's many kilometers? The ice is many kilometers thick.
That's a great question. So there's some material that is on the surface of Europa that that can be constrained through conductivity. There's material that was identified also, like salts, sodium salts, chloride salts, and sulfates. And based on the potential of all of that being part of the ocean, they create models that model the salinity. If you ask me, are we certain about the salinity? No. So when I said it's the same, it's the models. At this stage, we really need to hold on to those because it's the only thing we have. But the future missions, JUICE is already on the way from European Space Agency. Europa Clipper will be launched next year from NASA. And one of the main objectives is to characterize the surface to the point that maybe if we find some materials that we are sure of from the ocean, we will have all of that data from direct analysis. Yeah, thank you.
All right, we're gonna just check Zoom real quick to see if we have any Q&A questions, maybe from a very certain Lucas Lawrence, or maybe not. We'll see if he kept his streak of 3 lectures. I've completely lost us. There we go.
Thank you.
All right, well, it wasn't a question, but we did see Lucas Lawrence, our very own. So yeah, I'm going to just give my little spiel at the end. If you guys have been to one of these before, you know what's about to happen. Oh. Lucas has a question.
Oh, who's this person? It looks like he knows what he's talking about. Hi, Lucas. Thank you so much for the question. Yes. So depending on the host star, there's some modeling can be done that we can not predict, but we can make plausible assumptions of the type of pigments that can be dominating the planets, depending on the host star. I hinted a bit about the M stars, for instance. M stars have less luminosity than than sun stars, sun-like stars. That would be a problem. And this means, and also it has a black body shifted to the red, while on the sun has a black body right on the yellow. This means that photosynthesis on planets surrounding those stars will be more likely, the pigments will be shifted to the red as well, because that's where you're going to have the most light that you can use. And light shouldn't be a limiting factor for a good photosynthesis to be proliferative. So we might find organisms that have photosynthesis happening instead of the peaks being on the blue and the red. They'll be on maybe the orange and the infrared. Yes. And we have organisms here on Earth that actually do that. The ones, for instance, that are in the thermal vents, the ones that are in the bottom of lakes, or in dark zones on the lakes, like they can be in dark zones not because they are in deepness, but because they have coverage of other organisms, for instance. Those organisms are called the green sulfur bacteria. I understand the joke. They are green, yes. But some of them are also purple sulfur bacteria. And both of them have pigments that are shifted to the red. Some of them actually make the photosynthesis in the infrared. Those would be nice candidates for planets around these M stars. And the pigments are not chlorophylls, are bacterial chlorophylls, which are pigments that we can't see because, again, they are in the infrared. But the spectra loves them. The spectrometers, they love them because they are very reflective usually. So those would be, for instance, the pigments that we would look for on M stars. Other stars, I don't think there's work on it yet. But I would say that if it's a very radioactive environment, you would need a carotenoid instead of a bacterial chlorophyll. You would need an orange carotenoid or a black carotenoid, because they shield the organisms very well. Yeah.
Lucas continues his streak. Very good. So just to close up, thank you once again all for coming, and thank you, Dr. Coelho, for having, speaking to us and talking to us about pigments and all this wonderful stuff. Just in case anyone here is new, this lecture series is hosted by the Cornell Astronomical Society. We're an undergraduate organization here on campus. We run the Fuertes Observatory, which is up by McClintock Hall across the road, if you're ever interested in coming. We're going to be heading there right now. We have open houses every 8 p.m. to midnight on Fridays, except if there's like horrible weather, like a blizzard, we're there, even on cloudy nights like tonight. So, and we have one more lecture next week, same place, same time, and then we take a two-week break in the lectures, and then we finish up with our normal lectures on November 3rd. And then we have a super special lecture. It's still secret and special, so you don't know about it, but you do know about it, but you don't know about it. And that's going to be after that one. So basically, long story short, you like this lecture, we got another speaker next week, and then in 4 weeks, we have a final speaker. So yeah, thank you all for coming. Hope to see you at the observatory.